Hi, everyone. Thank you all for attending. And I also thank the organizers for inviting me. My name is Raquel Peixoto, and I will talk about restoring and rehabilitating coral microbiomes. I'm very sad that I cannot be there with you guys, but I'm glad that I can still present some of our data. I'm sorry. Yeah. We are all victims of the shifting baseline syndrome, which is the perception that the state experienced by the previous generation is or was the pristine state. Although in reality, and in most cases, it already represents a disturbed one, that most, if not all ecosystems are no longer pristine. So anthropogenic impacts and climate change are turning formerly pristine ecosystems and microbiomes into ever-changing new normal states. And in this sense, this biotic process, which are the breakdown of symbiotic relationships between the host and its associated microbiome, are very important triggers of environmental disturbance and outbreaks. The way it works is that healthy organisms and ecosystems rely on healthy microbiomes, just like all of us. And this healthy microbiome contributes with nutrient uptake and cycling, control of pathogens, mitigation of toxic compounds, and many other beneficial roles. The problem is that the beneficial microbes can also be, and usually are, sensitive to impacts and changes in environmental conditions. And what happens is that they are usually replaced by pathogens. So degraded ecosystems can become pathogen spreaders, which is an additional problem. And these dysbiotic assemblages are also one of the causes of current increase in the emergence of human pathogens. And the consequent realization that we need alternative and maybe more ecological solutions to cope with human disease. And of course, we are also applying these to biodiversity loss and any other application that involves microbiome stewardship. So what we think is that overall, if we make the organisms stronger, any organisms, they can virtually cope with any impacts. It's just like us. If we are healthy, it's less likely that we will be affected by infectious disease, right? But how to retain the native beneficial assemblages or rehabilitate beneficial assemblages rather than allowing pathogens to establish? This is the challenge, right? So there are innovative approaches that can restore and rehabilitate microbiomes to protect organisms and ecosystems that will ultimately support One Health, which is the concept that humans, other organisms, and environmental health are all interconnected. If we bring this into the marine realm, one of the most threatened marine organisms are corals, as you know. And in this sense, microbiome restructuring towards a more pathobiomic or dysbiotic assemblage is hypothesized to be one of the main causes of coral disease, and it's also strongly associated with coral bleaching. Therefore, one of the approaches that have been proposed to rehabilitate degraded reefs through coral health improvements is the use of beneficial microorganisms for corals, or BMCs, which are probiotics specifically designed for corals. And this is not new. The use of probiotics has been applied. Probiotics have been applied across different hosts, including wildlife, for example, bats, frogs, and bees. And in some cases, it can be the only medicine to cope with biodiversity loss. In the case of corals, what we did is that we proposed a parallel to plant and human probiotics by looking into specific traits that can be beneficial for corals, considering the coral physiology. So we select coral probiotics, or BMCs, that are native microbes associated with corals and very abundant in the marine ecosystem. And we isolate and assemble them into microbial consortia. trying to combine several of the putative traits that we described in our framework here. With that, we want to, one, tackle the cascade of different problems that are triggered by dysbiosis, and two, increase the chances that at least one of the probiotics used will be competitive against the opportunistic microbes. So we use a combination of microbes to increase the chances that at least some of them will establish, and also to tackle different problems that are triggered by dysbiosis. We use, for example, probiotics that can mitigate toxic compounds that are correlated with coral bleaching. For example, ROS, reactive oxygen species, and DMSP. So we use very good ROS scavengers and DMSP degraders that can mitigate these compounds. And in the case of DMSP, for example, dimethylsulfonylpropionate, this key component of the sulfur cycle is abundant within the coral holobiont, but increases in concentration when corals are stressed. And in high concentrations, they become a chemoattractive to pathogens, especially Vibrio. So when we degrade DMSP, on top of providing corals with an additional source of nutrients, we can also try to block the colonization by pathogens. And after we proposed this framework and these ideas, we test this concept. We proved the concept that probiotics could mitigate bleaching, at least in lab trials. And we moved beyond. We wanted to advance the research to try to understand whether we could also prevent coral mortality. And also we wanted to explore the underlying mechanisms associated with protection provided by probiotics. So in this study, we demonstrated BMCs triggered a dynamic microbiomes restructuring that was actually instigated genetic and metabolic changes in the coral host that ended up mitigating coral bleaching and mortality. So what happened is that corals inoculated with placebo either died or presented very low FVFM rates, which is an indirect health proxy that measures the photosynthetic ability of the symbiotic algae. Whereas corals inoculated with placebo all survived and presented very high FVFMs at the end of the experiment that were comparable to the rates observed in corals that are not even exposed to stress. We also described the post-heat stress disorder that is associated with coral bleaching by evaluating the up and down regulation of specific genes, and that this disorder was mitigated by BMCs. And our metabolomic and metatranscriptomic data indicated that this protection was signified by patterns of DMSP degradation, which was one of the mechanisms selected in our consortium, lipid maintenance and a coral host transcriptional reprogramming that included cellular restructuring, repair, stress protection, immune genes, and the consequent increase in coral survivorship and a stable FVFM in protected corals. Since then, we and many other authors have been expanding this type of application to use different types of probiotics to cope with different impacts, different threats, and also used at different life stages. So we have tested probiotics for different coral species using different microbial assemblages in lab trials, and this application has been always successful. So now what we are trying to do is that we're trying to move forward to perform the first pilot trials to protect corals against thermal stress in the real world. And for that, we established the first coral probiotics village, which is an underwater facility that is well and intensively monitored, where not only biotic, abiotic, and risk assessment factors are monitored, but we also monitor different organisms and their microbial connectivity and health physiology. So we want to evaluate the efficiency of coral probiotics to improve coral health and go beyond. We want to know how the One Health concept applies to that. We want to know how other organisms respond to this inoculation. And of course, we also want to see if there is any potential harm that can be caused. So What we want to know is whether these probiotics will cause any harm, or they will protect corals, and consequently they will protect other organisms. So with that, we are evaluating whether we can move towards a whole reef restoration concept where probiotics could benefit corals and other organisms. As in the end of the day, what we are doing is that we are retaining what was there before and blocking pathogens to overgrow. And this applies and this connects to One Health. And while we do that, in parallel, we are trying to develop ways to upscale this type of application should it prove these efficient and safe. So we are trying to develop tools that can facilitate the application of probiotics in the reef. For example, we are using AI and robotics to scale up and facilitate the application of probiotics. Using these automated irrigation systems that can inoculate corals in the field and can be regulated from apps in our phones. In the coral village, we are also there are also other PIs working on different solutions and technologies like 3D printed corals and the use of manufacture. And some of the people involved in this work are actually in the conference, like Professor Charlotte Hauser and Sebastian. I don't know if Carlos is also there. But the good thing about the Coral Probiotics Village is that we can integrate all of these different solutions and try them for different applications, and also combined, and at the same time combine these with different expertise and monitor the response of the reef as a whole. And for example, we are also testing how coral probiotics could be applied to coral nurseries, different types of nurseries, how the deployment would be performed, and whether these probiotics could also make corals grow faster and in a healthier way, like that they could be more resilient at the end before they are outplanted. And to facilitate implementation and pilot tests that we are developing, we have recently proposed a framework that is science-guided and flexible and can inform stakeholders and scientists regarding the need for risk assessment steps that can at the same time ensure the safety of this approach while also allowing the development of these urgent science. And this type of framework can be expanded and adapted to weigh the use of different technologies against the high toll of inaction. That is a decision realistic framework that considers what's happening in the real world if we don't do anything, rather than only theoretical fears that are based in pristine ecosystems that no longer exist. So in this case, in the case of probiotics, we propose that this should be a case-by-case decision, that we should consider alternative treatments if they are available or not, and if they are not, we should also consider the risk of inaction, and of course follow the very strict safety risk assessment steps that can guarantee that we minimize any potential impact. And this type of framework can be also adapted to different solutions that should be applied for coral reef restoration, and the fact that we need to weigh the high risk of not doing anything. And there is no magic bullet, of course. And these solutions, in my opinion, they will need to be applied in combination and depending on the reef that we are trying to restore. So to recap, the mitigation of this biotic state of marine and the marine degradation needs to consider this shifting baseline syndrome or trap, where we are trapped in as the outset for restoration and rehabilitation, and not pristine ecosystems. And that this restoration is a parallel to other types of restorations that are very well accepted, just like fecal transplants in humans, or the restoration of forests. So you replant degraded forests, right? So we need to replant degraded marine microbiomes. It's just a matter of retaining what's beneficial and avoiding pathogens to overgrow. And we have being enriching pathogens with anthropogenic action. And it may be our moral obligation to try to retain beneficial microbes that are easier to manipulate than the hosts in many cases. And so this manipulation can be very effective in some cases. We need to try to retain the good ones and avoid pathogens to spread. So overall, in my opinion, uh, restoring marine microbiomes is no longer a decision that we will need to make. It's a reality we need to face, and then we need to decide how to better do that. And this is definitely something that coral restoration should consider any approach. With that, I thank my team, the amazing people behind all of these data that I presented to you, and I also thank my sponsors, my collaborators, and you all for attending. Again, it's a shame that I'm not there, but I'm very happy that I've managed to present some of the work we are doing. Thank you very much.